Hello, everyone, and welcome to another episode of Nerd RX podcast. Today, we have immunohistochemistry as the topic, and to talk more about it, we have Dr. Shveta Parekh. Welcome, Shweta, to the show.
Hi, how are you?
I'm good. How are you doing?
I'm doing good.
Well, thank you so much for being with us here today. And before we jump into the topic, we would love to hear about your journey and your background and how did you get to this point in your career?
I am currently a teaching assistant professor at the University of North Carolina, Chapel Hill. So I would say I had a pretty kind of traditional background. I am originally from New Jersey. So I went to Rutgers University, which is a state school in New Jersey, for my undergraduate degree. And I double majored there in genetics and psychology. And it was here that I really fell in love with research. I was able to be part of research as an undergraduate research assistant. And that's what really led me to my kind of career and wanting to apply to grad school. And so I applied to graduate school here at University of North Carolina, Chapel Hill, and received my PhD in that program. And during my experience in graduate school, I was able to teach a undergraduate course. And I realized that through that, teaching became one of my passions. And that led me
Hmm.
to pursue my faculty position here at UNC that I'm currently working on.
Wow. And are you enjoying your faculty position currently?
Yeah, I love it. I really do enjoy teaching. I'm currently in the neuroscience department,
Hmm.
so I'm really enjoying teaching neuroscience courses to undergraduate students.
Well, that's awesome.
Thank you.
Talking about IHC, immunohistochemistry, um, why did you pick this topic and what got you interested in it?
Yeah, so immunohistochemistry, or IHC for short, it's actually a really old and basic technique.
Mm-hmm.
And it was actually invented over 120 years ago. And I spent a lot of time in graduate school using this technique to kind of answer my research questions. And I really got interested in it because although it's a very basic technique, nowadays using different types of antibodies and high throughput microscopy, we're able to really answer a large variety of different research questions using this very basic technique.
Mm-hmm.
And so
Okay.
specifically, yeah, in my like graduate school, I used IHC to visualize proteins
Uh-huh.
on cells in brain tissue of rodents.
Okay. And you used rodent tissues in your research?
Yes, yeah. So my research focuses on heroin withdrawal. So we
Mm-hmm.
were specifically looking to see what types of cells and cytokines are involved in heroin withdrawal. So we
Mm-hmm.
would use very thin slices of rodent tissue and then we would conduct IHC on those thin slices of tissue to be
Mm-hmm.
able to see these different kind of chemicals that we're interested in looking at.
Mm-hmm. Okay. So, would you just give us an overview of various steps involved in this process of IHC?
Yeah, so basically, with immunohistochemistry, the first thing is you need a very thin piece of tissue.
Okay.
So once you have that thin piece of tissue, you can normally we use a cryostat, which is a type of machine that you can like slice that tissue. Um, I almost like to relate it to when I teach my students about it. I relate it to a deli slicer, so something that slices meat really thin.
Mm-hmm.
But it's basically the same thing. We use it for brain tissue in this case. And so then after that, what you're going to do is you're going to apply kind of a bunch of different buffers to the sample to prepare it. And then you use antibodies, which is the most important aspect of immunohistochemistry. So the first thing you do is you apply a primary antibody, and
Mm-hmm.
this is an antibody that's designed to attach to the marker that you're interested in. So that primary
Mm-hmm.
antibody is going to go on that thin slice of tissue. and it's going to attach to whatever marker you want to look at. And then the secondary antibody, that one is going to go and attach to the primary antibody.
Mm-hmm.
And what's really interesting is that the secondary antibody will have a dye that's attached to it. So this goes back to kind of how it's such a basic technique. So originally, these dyes would be stained pigments that were
Okay.
attached to the antibody. And so you would use light microscopy to visualize your protein. However,
Mm-hmm.
nowadays, the more common technique is that these antibodies have a fluorescent dye that are attached to it, and so you can look at your sample under a fluorescent microscope, and wherever your marker is, it glows, and it really creates these beautiful images.
Mm-hmm. Yeah, I think IHC has one of, when you look at a paper, IHC gives you some very beautiful images to
Yeah, I
publish.
yeah, I think my favorite part of doing it is really seeing that tissue under the
Mm-hmm.
microscope because and then what's really fun is you can use different types of fluorescent dyes. So you can one fluorescent dye can be red, another one can be green, another one can be
Yep.
purple, and then you can put them all on the same tissue and really have all of these variety of colors.
Mm-hmm. Yeah, right. So in one of my previous episode, we were discussing a similar technique, and they told us that one of the step that is very important is permealization.
Mhm.
So is that step also involved in IHC?
So it can be, it doesn't have to be. The
Okay.
beauty of IHC is that it really depends on what you're looking at. So if your kind of marker in that tissue
Mm-hmm.
is really low, kind of not prevalent that much, and you really have to go into the tissue to have that antibody attached to that marker,
Mm-hmm.
then it can be really important. But
Okay.
if you have an abundance of your marker, then it's not as important. So it really
Mm-hmm.
depends on what you're looking at.
Okay. Okay. Yeah, that makes sense. And can you also use, for example, multiple primary antibodies in one tissue to, for example, see co-localization of some proteins?
Yeah, so that's exactly what I was doing in my research. So, what I was specifically looking at, I was interested in seeing whether cytokines, so cytokines are markers that can increase or decrease following inflammation. So, I
Mm-hmm.
was interested in seeing where these cytokines were coming from. Like which types of cells. And so within my really thin slice of rodent tissue, I applied multiple primary antibodies. So I would apply
Mm-hmm.
one that would attach to neurons, one that would
Mm-hmm.
attach to astrocytes, and another that would attach to microglia. So those are all three different types of cells that you can find in the brain. And
Yep.
then I would also apply another antibody that would attach to cytokine. So the cytokine in particular that I was interested in looking at was interleukin 1 beta.
Mm-hmm.
And so when you do all this, you're really able to see co-localization. So I can see, how much interleukin 1 beta is on the neurons, how much of it is on the astrocytes, and how much of it is on microglia. And you can do that through co-localization.
Okay. Okay. So this definitely makes it more versatile, and you can use multiple things. So that
Yes,
is
yeah.
awesome. Hmm. So are there any alternative techniques to IHC that would give you similar data what you get in IHC?
Yeah, so there's a couple, and I think maybe it doesn't actually give you the exact same data, but I can talk about kind of the differences.
Yeah.
So when we're talking about IHC, which is the markers that we're talking about are related to protein. So the antibodies are tagging proteins in that tissue.
Hmm.
So there's a couple other techniques that you can use to quantify protein. So one would be Western blot.
Hmm.
But in that technique, what happens is instead of a very thin slice of tissue, you kind of have to mash all that tissue together,
Yeah.
and it really gives you a good amount to be able to quantify the protein. So you're able to say,
Correct.
is this protein present in the tissue, and how much of it is there. But what
Mm-hmm.
happens is you lose spatial recognition. So in
Right.
IHC, what's so nice and I really like, because I'm a visual learner, is you
Mm-hmm.
can see where all of these different proteins are located within that slice of tissue. With
Right.
Western blot, you're not able to do that.
Correct.
Um, yeah, so I think that, there's some give and take there. I think with
Mm-hmm.
IHC, you're not really able to, the quantification gets a little bit messy
Mm-hmm.
because it's only a semi quantification.
Yeah.
Western blot has a more proper form of quantification of that protein, but you're not able to get that pretty picture. You don't see those pretty
Yeah.
fluorescent cells, and
Mm-hmm.
you can't really see where those cells are or how the
Yeah.
cells are kind of in structure, right? So with
Mm-hmm.
IHC, what's really cool is when you stain, you can see that these cells might not look as great or might not look to be what you
Mm-hmm.
expect to be. And that might give you an indication, maybe these cells are dying. It's possible that something happened. With
Mm-hmm.
Western blot, you don't get that
Yeah.
image, so you really
Mm-hmm.
lose spatial recognition.
Wow, that's a very good way of looking at it. And you mentioned that IHC is not as good in quantifying proteins, but it tells you where it is located and the state of a particular protein. Um, so, do you have to always run, in order to publish, mostly, do you have to run a second technique to kind of confirm your findings?
So in a lot of cases, you will see that people who do conduct IHC will run a Western blot, or they might run qPCR. So
Mm-hmm.
qPCR is another technique that might be an alternative to IHC. However,
Mm-hmm.
in qPCR, you're really looking at that mRNA, which
Yeah.
is a precursor to the protein.
Yeah.
So a lot of it is common that when people publish, they do IHC in addition to Western blot or qPCR,
Okay.
because both of those techniques don't have that spatial recognition but have
Mm-hmm.
high quantification. But it's not
Mm-hmm.
always the case that you need it. So I know I've personally published without both
Okay.
of those techniques. So I think something to confirm your effects is you can use different programs to kind of calculate the percent area of that
Mm-hmm.
protein in any given space. So that space would be
Okay.
your tissue.
Okay. Awesome. And would you say IHC is user friendly and anyone can learn it easily without having any background in it, or does it have a huge learning curve?
I believe so. The beauty with this technique is I really think it's very user friendly. So
Mm-hmm.
there is, of course, a little bit of a training period.
Yeah.
Um, However, I know that I've successfully trained multiple students, undergraduate students at UNC, um, immunohistochemistry, as well as at UNC, we have an undergraduate neuroscience laboratory course that teaches immunohistochemistry to sophomores and juniors in
Okay.
their kind of college degrees. And these students come in with kind of, they have a conceptual background of how it
Yeah.
works. So a little bit of what we're talking about today, but no real laboratory skills. And,
Yeah.
by the end of the semester, they're successfully conducting IHC on their own and getting beautiful images on a
Mm-hmm.
microscope.
Oh, wow. So, what would you say is in this entire process? You made it sound so simple, but according to you and in your experience, which step is requires the most amount of troubleshooting, or kind of can mess up your entire experiment?
Yeah, so I think with immunohistochemistry, the specification of that antibody can be very difficult. So one disadvantage with immunohistochemistry is that you really can only use this technique with developed antibodies unless you want to go and develop your own.
Yeah.
Um, and with those developed antibodies, It really depends on how long that tissue is sitting with that antibody on it, or how long that tissue is kind of soaking in the antibody mixture. And that can involve a lot of troubleshooting. So
Mm-hmm,
sometimes there are some antibodies that, you can get a beautiful stain, and it only needs to be on the tissue for an hour. And other
mm-hmm,
antibodies don't work as well, and so in order to really pick up that protein, they need to be on the tissue for overnight kind of incubation.
mm-hmm,
And so that kind of figuring out how long that antibody needs to sit on the tissue is the
mm-hmm.
hardest step.
Okay, so before going into your actual experiment, you have to run a bunch of trials.
Yes, yeah, a lot of
Mm-hmm.
trials.
Right. Okay. Okay. Yeah, that is, I think, with most of the techniques, even Western blot I'm doing, I'm running a bunch of trials from
Yes,
like months now.
yeah, it can. I mean,
And,
even one antibody can take that long, which is why
mhm,
that can be the downside to this technique. It takes a long time to kind of perfect that timing, but
mhm.
once you have that timing perfected, it works every time.
Yeah, it becomes like a protocol in your
Yes,
lab,
yes,
and no one else has to spend that time again doing all those things. So I think in
exactly.
the end, it's worth it.
Yeah.
So, in your experience, has it ever happened to you that you know how we buy antibodies from these companies, and most of the time they mention and they reference so many papers, and you go and look at it, oh my god, they have this beautiful images, but when you do it, it doesn't work.
Yes, I think that that's definitely happened to us. When I was a second year in graduate school, I kind of didn't really understand this whole troubleshooting process. I
Yeah.
was a little naive, and I thought to myself, these companies, they sell us these antibodies and they have this whole protocol on their website.
Yep.
I don't need to troubleshoot. I'm just going to go ahead and
Uh-huh.
use their protocol.
Yeah.
And it doesn't always work, and it didn't
Hmm.
work for me. And I think
Hmm.
that's because everyone kind of uses their tissue or prepares their tissue slightly differently.
Yeah.
So I know that a lot of things with immunohistochemistry, it can really depend on whether your tissue is frozen or fresh.
Hmm.
And so when I do immunohistochemistry, I prefer to use fresh tissue.
Okay.
So that means that once it's taken out of the animal, it hasn't been frozen. It's just been kept at a low temperature in a buffered solution.
Okay.
But a lot of people choose to use frozen tissue. So they
Yeah.
will freeze it at -80 or -20 and then conduct IHC. And either way works. But then you really need to pay attention to that antibody, because certain
Mm.
antibodies work better on frozen tissue and certain
Oh, wow.
antibodies work better on fresh.
I did not know that. That's a very good point. I have done IHC a bunch of times, and it's a little pain sometimes. But
Yeah.
I think the worst part of IHC is putting the tissue on the slide.
The
That
mounting
is,
process.
yes, the
Yeah.
mounting part. Yeah, that I do not enjoy. And we had this last step remaining to for a paper. They had asked us to do like a follow-up study. I had like one day, and I was mounting like 150. That was tiring.
Oh, no, I would never do that. I limit myself to when I have to mount. I It's
Uh-huh.
also not my favorite process either. I
Yeah.
make it a point only doing it for 2 hours, otherwise I get
Uh,
frustrated.
yeah, that day I sat for like 6 hours straight
Oh
just
gosh,
mounting.
I would go insane.
Yeah, that's something which I don't enjoy, but it does give very beautiful images,
Mhm.
so I think it's worth it in the end.
Yes.
Just I just got this question. Like in Western blot, whenever we get a new antibody, the first thing we do is obviously figuring out the dilution we use
Yes.
for our blots. Do you also have to do that in IHC?
So yes, you can change your dilution depending on what kind of tissue you're using. For the most part, it really depends on how you're doing IHC. So there's two types of ways you can do it. One is called slide mounted. So that's where you mount your tissue first. And
Okay.
then you apply all these buffers and antibodies in very small quantities directly on the tissue.
Hmm.
So in that case, those dilutions are going to be very, very small.
Yeah.
In the other case, it's called free floating IHC.
Yeah.
So that's
Hmm.
where you have all your tissues kind of sitting in a dish or a beaker, and you
Hmm.
apply a large amount of solution to that beaker. So the tissue is literally floating in
Hmm.
that liquid, and that liquid contains your antibodies. So
Hmm.
for that type of scenario, the dilutions of the antibodies can be larger. So
Okay.
you can kind of imagine that there is kind of a benefit to one over the other. So the
Yeah.
free floating, you're going to be able to stain that entire tissue and really
Correct.
get a great image of the whole area.
Hmm.
Um, However, you do use more antibody in that case.
Yes.
And in this process, the antibody is the most expensive.
Correct.
And so, if there's any way to reduce the amount of antibody you're using, that can be more
Hmm.
cost effective.
Hmm. Yeah, to, like you mentioned, antibodies are the most costly of this entire process. Even in my Western blot, I'm trying to use like the smaller and smaller incubation chambers
Yeah.
so that I bring down the volume and the amount of antibody I use. So I
Yes,
totally get
we
that.
definitely think about that in terms of it's kind of the give and take on, do you want a better picture or do you want to
Right.
save money? So you have to
Hmm.
find that nice line between the two.
Correct. So, what would you say are the advantages and disadvantages? I know you mentioned a few disadvantages, but just in general, are there any more?
So I think for disadvantages, I kind of mentioned that the quantification isn't
Yeah.
exact, and the specification of the antibody can
Mm-hmm.
be difficult. And I know that we touched upon this, but these stains are not standardized worldwide.
Okay.
So there's no machine doing these things, right? So it's a human that's applying all of these solutions to the tissue and taking
Mm-hmm.
images. And so of course there's going to be human error that's
Yes.
involved. And so a lot of times it can be frustrating when you see a protocol online or in a paper and it doesn't
Yeah.
work exactly the way you want it to in your own hands.
Yeah.
But
Yeah.
I think this technique, I mean, I think some of the advantages are that, these tissue samples can be stored for such a long time.
Right.
So you don't have to immediately use the tissue. You can kind of take that tissue and store it and look at
Yeah.
it later.
Mm-hmm.
It's also very affordable. So this is
Mm-hmm.
it's pretty cost effective. And it's
Mm-hmm.
one of the cheaper techniques in science because it doesn't involve expensive equipment. You really just
Mm-hmm.
need that tissue, your microscope slides, buffers, antibodies, and then the microscope to look at the tissue under.
Yeah.
And so the most expensive thing, like we talked about, is the antibodies themselves.
Antibodies.
And
Okay.
you really get a great spatial localization of whatever you're looking at.
Mm-hmm. And do you use confocal to image these?
Yeah, so we use both a bright field and a confocal microscope. And of course, the confocal microscope gives you such a better
Yeah.
image.
Yeah.
But the nice thing is, you don't need it, right? You can use a
Mm-hmm.
bright field. And even with some IHC techniques that aren't fluorescent based, you don't even need a fluorescent microscope. You can just use a light
Right.
microscope, which are very, very
Okay.
cost effective.
Mm-hmm. Okay, yeah. I think you covered my next question about the
Oh,
cost. So that's awesome. So it's good that I think any lab just working with tissues can set this up easily. So, like you mentioned just now about storing the brains, how thin do you slice each section?
So for if you're doing a slide mounted
Mm-hmm.
immunohistochemistry protocol where you mount the tissue first,
First.
that has to be generally it's a 20 micron slice or
Mm-hmm.
thinner if you can get it. But for free floating, you can do it anywhere from 40 microns to 100 microns thick, depending
Okay.
on what you are really trying to look for.
Yeah. So from one brain, you can get so many slices, and it's not like you will run out of the
Mm-mm.
sample so quickly. That is another.
Exactly. Yeah. Yeah, that's a great point because when we run an experiment, it's possible that we have this hypothesis in mind. And so we
Yeah.
conduct that first stain. But once we
Mm-hmm.
look at that stain under a microscope, we're able to see, hey, maybe this might work and this might work. And it's really great because I won't have to rerun that experiment with
Yeah.
the animals. I just have the tissue to begin with. And so I can just run another stain and another stain and
Yeah.
look at that under the microscope as well.
Yeah. Yeah. Okay. So, are there any fun facts about IHC?
Yeah, so the history of immunohistochemistry, how it was invented, I think is a really interesting story. So there
Mm-hmm.
was a German physician, Dr. von Behring, and
Mm-hmm.
he actually received the first Nobel Prize in physiology and medicine in
Wow.
1901. So what he did was he discovered diphtheria and tetanus antitoxin. So he was mostly known as this huge savior of children because back
Mm-hmm.
then diphtheria and tetanus used to be really big causes of child death. And so, what he did to research these antitoxins, where he would inject diphtheria and tetanus toxins into guinea pigs, goats, and horses. And eventually, these animals would develop an immunity to these diseases. So, they were able to kind of protect themselves from getting sick.
Mm-hmm.
And so, what he would do is he would look at their blood, specifically their serum, and he would derive these antitoxins. And so, these antitoxins could protect people and cure the diseases in animals and humans. And what's really interesting and how this kind of relates back to immunohistochemistry is those antitoxins actually contained antibodies.
Okay.
And so through this process of him kind of discovering these antitoxins, that's how these primary antibodies were discovered. And that's exactly these primary antibodies that we use today in immunohistochemistry.
Mm, that is so interesting. Like
Yeah.
going back so long. Oh, wow.
It's a really old technique and
Mm-hmm.
I think that that's the beauty of it. It was actually
Yeah.
discovered accidentally.
Wow. Yeah. Those techniques are something special. Like when you look at, all the history of things, there are so many important discoveries done by mistake.
Yeah.
And that is like really awesome.
Yeah, and I think it's really cool because now, that it was discovered by mistake and they used it to like clinically, they use those antitoxins to treat people who were sick, but now clinically immunohistochemistry is huge in the field of cancer,
Yes.
specifically as a diagnosis tool.
Mm-hmm.
So, I know when they're looking for specialized tumors,
Yeah.
they use immunohistochemistry because each tumor in your tissue is different. So those tumors are going to express different types of antigens, which is a
Mm-hmm.
particular marker that we can stain in
Mm-hmm.
immunohistochemistry. And
Mm-hmm.
so it's amazing the information they're able to gain. Clinicians can use them in immunohistochemistry and
Right.
they know which type of tumor is present, the stage, the grade of the tumor, as
Yeah.
well as like where that tumor originated from. So if
Mm-hmm.
possible the tumor came from the liver and now it's found somewhere else, they can kind of find that site of origination.
Mm-hmm.
And I think what's really interesting, I was reading a paper the other day is that now they're able to use immunohistochemistry to kind of predict therapeutic responses of tumors.
Mm-hmm.
So they're using it to understand better what type of treatment will best target that type of tumor. So
Mm-hmm.
before they even kind of start that treatment for
Yeah.
cancer patients, they'll take samples of the tumor and use immunohistochemistry to apply different types of treatments and see which ones are kind of better targeting that tumor. And that's
Wow.
going to be the one that they now use.
Oh my God, that is so cool. I
Yeah, I
did
think
not
that's,
realize IHC could do that.
yeah, I didn't either. I thought that that was amazing.
Yeah. Well, my last question for today is, could you suggest an interesting article which you really enjoyed reading about IHC?
So there's this journal or kind of like a magazine called Scientific American Mind.
Mm-hmm.
And they have this one article. It's really a slideshow of slices of brains that have been stained with IHC, and they
Mm-hmm.
have these really beautiful images. And the article name is called I think it's called Beautiful Minds. Imaging cells of the nervous system. So it really shows you what IHC is capable of, and they have
Mm-hmm.
brief descriptions under those images to kind of explain what you're looking at. And I really enjoyed reading that.
Okay, I'll make sure to have that in my description linked
Yes.
so that people could definitely go and look at those beautiful images. Uh, well, Shweta, this was a very, very informative episode, and I did not realize what IHC could do. So, thank you so much for that.
Yeah, of course. Thank you so much for having me. This was a lot of fun.
It was my pleasure. And listeners, I will catch you next week on another episode of Nerd RX podcast. And in meanwhile, if you have any suggestions or if you would like to come and do an episode with me, please email me at barkha@nerdrxpodcast.com. And remember, it's good to be a nerd. Bye.